this is what we are going to explain today. This is going to be a replication, okay? The process of replication. As I told you before, replication is the process that happens before a cell division. I don't care if we are talking about a mitosis or a meiosis, and it happens in the interface. If you remember, it happens in the S phase of interface. This process is essential for what? For keeping the same genetic material all the time and have no problems and no mutations for example, when you do a mitosis. So it's a process that is really, really important. So this process is going to happen both in eukaryotic cells and prokaryotic cells. In eukaryotic cells, it's going to happen where? Inside the nucleus, right? Because we have a nuclear envelope. And in the prokaryotic cells, it's going to happen in the cytoplasm. Okay? And as I told you, it happens before cell divisions, mitosis or meiosis. How it happens? Imagine that you have the double strand. I have paint one in red, the other one in black to see the differences. Some enzymes, enzymas, are going to break the bonds that join the two strands. Remember, the two, if we have a thymine here and we have an adenine here, they were joined by two hydrogen bonds. So there are some enzymes, helicasas, topoisomerasas, they have different names that I don't need you to know, that are going to break the bonds and open that strand like this. Because if not, you don't have enough space to do another strands, because you have to do the strands like this. So you need a space. So these enzymes are going to work and they are going to create a replication bubble. Okay? And how are they going to, how this replication is going to happen? When you separate these two strands, I have put them like this. This is the black one and this is the red one. You have a sequence of nucleotides, right? I have put this one, for example, adenine, thymine, cytosine, guanine, thymine, whatever. And the other strand, remember, due to complementary bases, they have to be the complementary bases. So it has to be, if you have an adenine here, you have to have a thymine here, right? If you have thymine here, you have to have an adenine here. Yes? Understood? So you have these two strands that are the old ones. And you are going to synthesize another one. No? I told you, the new ones are going to be here and here. So we are going to use one of these ones as a model, no? The old ones as a model, and you are going to create a new strand. This is called semi-conservative process, because when you finish with the replication of the DNA, you are going to have one of the strands is going to be the old one, and the other strand is going to be the new one, right? And here exactly the same. You have the old one, and you will synthesize the new one. Yeah, understood? Okay, so how is this going to happen? How are we going to create a new strand? It's going to happen because we are going to have some enzymes. In the case of eukaryotic cells, it's going to be called DNA polymerase, ADN polymerasa in Spanish, and this is going to be the enzyme in charge to produce the new strand. Okay? Again, with complementary bases, you are going to create the new one. Okay, so you have here adenine, you have here a nucleotide with thymine. if you see, I didn't put the phosphoric acid and I didn't put the deoxyribose, the sugar, because everywhere is going to be the same. So to simplify, I have put just this. Okay? And here exactly the same. You have the other strand, your old strand, and you're going to create the new one. Exactly the same. The DNA polymerase is going to create this new nucleotides and is going to join it in the position that this strand is going to tell. Okay, so indeed during this process it's very important not to have mistakes, because if you have mistakes, mutations occur, remember? And mutations can be good, yes, but they can be also not that good. And you can have problems with this type of mutation. So you have, all of us, we have repair enzymes that are going to go after the creation of the new strand, just to check, repasar, to check that everything has been done correctly. And if, for example, you have here, let's put it in red, no? A cytosine, which is a mistake, this repair enzyme, exonucleasa, is going to come here, is going to remove this nucleotide and is going to put the correct one. Right? More or less understood how they work? Okay, even having these repair enzymes, we can have mistakes. So if this mistake happens, yeah, a mutation will occur, okay, during the replication process. I think that basically this is all. If you have any question, just ask me. And now what I'm going to do in another video is to correct the exercises from the day before. Okay? Bye-bye.